Hello, welcome all to this third seminar of the Kalim Red Talk series. I am Bertrand Nedel, director of the Kalim Institute. I'm not going to bother you about it, but if you don't know our institute, I encourage you to go and type Kalim in your browser and enjoy our brand new beautiful website. As you may know, the Red Talk series were born from the desire to have a regular scientific event focused on hot topics in the field of lymphoma, an event which would bring together experts, not only restricted to our consortium, but in the contrary, open to all lymphomaniacs around the world in order to foster discussions, brainstorming, networking, collaborations, and so on. So Red Talks were born, and they are every first Thursday of the month, 6 p.m. CET. It's open access, it's free, you just need to register as you just did today. This is a webinar, so you won't be able to directly ask questions, but the moderators will do it for you. Just formulate your question through the question tab, chat in the chat tab. Don't do the reverse. Don't chat in the question and question in the tab. But normally all the questions should be addressed. Last point, we'll close the webinar at 7 p.m., but the Q&A session will, with the speaker will remain open for an extra half an hour, so you can ask all remaining questions. So I'm done, that's it. Without further ado, I let Sandrine and Karine introduce our speaker, Laura Pasqualucci. Many, many thanks, Laura, for accepting our invitation. Unmute, Karine, unmute.
Better. Okay, thank you. So, hello everybody. So I am Karine Tart. I am professor in immunology at Rennes Medical School, and I also had a research unit, an INSERM research unit dedicated to the biology of B-cell lymphoma, with a specific interest for the crosstalk between the tumor cells and the microenvironment. And it's really my pleasure to be here today to welcome your questions and try to organize all these things. And now it's time for Sandrine to present herself.
Yes. Thank you, Karine. So my name is Sandrine Rouland. I am a researcher at INSERM, and also I'm leading a team at the Center of Immunology of Marseille Lumini, focusing mostly on the pathogenesis of B-cell lymphoid malignancies. And so, of course, it's also a great pleasure to introduce our speaker tonight for France, Laura Pasqualucci. So as a brief introduction, so Laura is a professor of pathology and cell biology at the Institute for Cancer Genetics, Columbia University. So she initially received her MD from the University of Perugia in Italy before moving to the US in Riccardo della Favera lab, where she started to be interested into the molecular genetics of lymphoid malignancies. And over the last, I think, 20 years, Laura's work and research team has made a major contribution into the understanding of the most common lymphoid malignancies, mainly follicular lymphoma and diffuse large B-cell lymphoma, discovering a lot of multiple lymphoma driving genetic lesion, and among them the discovery of epigenetic modifier genes that are frequently mutated in lymphoma, such as CREB-BP or KMT2D. And so more recently, our research group has been focusing on the understanding on how those mutations in chromatin and histone modifying gene are contributing to modulate the epigenetic landscape of the normal B-cell idiom in the germinal center reaction. And today I think she will talk maybe more in details on how those epigenetic modifiers modulate and contribute to the formation of those lymphoma tumors, and how we can potentially use and harness this knowledge in terms of therapeutic potential. So I think I will not give more detail. I think Laura will do it. And we are really excited to hear you, Laura, on that topic. And the floor is yours whenever you want.
Thank you very much, Sandrine. And thank you, Bertrand, and everybody for inviting me to speak and for being part of this series. This is really an honor. Now, as Sandrine just mentioned, I'm trying to set up the slide sharing here. As Sandrine just mentioned, the main research interest in our group is in understanding why and how B-cell lymphomas develop. And the way we have approached this problem has been traditionally through the identification of genetic alterations that are associated with these tumors, because obviously this would reveal genes and pathways that are presumably required for the survival of the cancer cell, and therefore would provide critical information in order to develop improved diagnostics or prognostic biomarkers, as well as, most importantly, for the design of targeted therapeutic approaches. So in the next 40 minutes or so, I'm going to first give you a broad overview of what we learned about the genetics of these tumors. I will focus in particular on diffuse large B-cell lymphoma, although many concepts apply obviously to follicular lymphoma as well. And then I'm going to focus on more recent studies that we have conducted and that we are conducting in the lab in order to understand why and how these lesions that affect epigenetic drivers contribute to malignant transformation. And now, I'm sure most of the audience would not need this introduction, but for those of you who do not think about lymphoma every day, a fundamental concept for the understanding of the pathogenesis of these tumors is the intimate relationship with this structure here, which is the germinal center reaction. Now, germinal centers are microenvironment with highly specialized functions that form transiently in peripheral lymphoid organs every time a naive B cell encounters an antigen for the first time in the context of T cell help. It's a highly dynamic environment where B cells constantly recirculate between these two distinct microanatomical areas that are also functionally compartmentalized. So one is the dark zone, where B cells proliferate at very high rate. In fact, this is one of the highest in the mammalian cells, with less than 12 hours duplication time, and also do something that no other cell in the body does, which is they somatically hypermutate their immunoglobulin genes in order to produce antibodies that have higher affinity for the antigen. And then the light zone, which instead is populated by more quiescent cells that make contact with follicular dendritic cells and then compete for help by a limited number of T follicular helper cells in order to be either positively selected to become an effector plasma cell or a memory B cell, which will maintain the memory of the immunizing antigen for life and can actually even be recruited again into new germinal center reactions, although this appears to be a relatively rare event, at least in normal conditions. And then there is a subset of light zone B cells that instead re-enter the cell cycle and can migrate back to the dark zone for multiple rounds of proliferation, somatic hypermutation, and selection in order to further refine their affinity for the antigen. But by and large, the vast majority of germinal center B cells are destined to be eliminated by apoptosis. Now, the dark zone and the light zone populations obviously are associated with distinct transcriptional programs and in fact, an even a greater degree of heterogeneity has now been recognized thanks to a number of single-cell RNA-seq studies, including the ones from Bertrand and Sandrine's group. This that you see here is a UMAP plot from our own data. It's human germinal center B cells, and yes, you probably know each dot represents one cell. Distinct colors would define diverse population of cells that share expression of the same gene sets. And so I think you can easily recognize the main stages of germinal center differentiation here, the dark zone, actually subsets of dark zone in blue, and the light zone B cells here in red. But there is also a very large number of intermediate populations, which may constitute more than 30% of all cells. And these would represent transitional states, as also shown by Bertrand, that continue circulating between these two areas. And then finally, we can see precursors to plasma cells as well as memory B cells. Now, the key point I want to make on this slide is that germinal center B cells must have a tremendous degree of plasticity in order to constantly and very rapidly switch between these distinct transcriptional states. So because consider that as many as 30% of light zone germinal center B cells have been estimated to return back to the dark zone every 5 to 6 hours. So this poses a significant stress to the cell, and together with the high proliferation rate, the replication stress, the DNA damage stress that is associated with the process of somatic hypermutation and class switch recombination, which I should say also take place in the light zone of the germinal center, may represent quite a burden, especially if you consider that these cells do not have proper DNA damage responses because genes like P53 or ATR are actively suppressed in this population by the master regulator of BCL6. So I would say it's not surprising that germinal center B cells are especially prone to malignant transformation, as demonstrated by the fact that the vast majority of mature B-cell malignancies, and you see here follicular lymphoma, diffuse large B-cell lymphomas, derive from a germinal center experienced cell. So from a transcriptional standpoint, the segregation of these germinal center functions is under the strict control of a complex network of transcription factors, which you see here, and signal transduction molecules that are also differentially expressed between these compartments and must be subjected to a tightly and coordinate regulation in order to guarantee this orderly execution of programs. So, for instance, MYC is required for the initial seeding of the germinal center reaction, but then its expression is suppressed during the majority of germinal center B-cell states, only to be reactivated specifically in this subset of cells that re-enter the cell cycle and migrate back to the dark zone in what is known as cyclic re-entry of the germinal center. On the other hand, genes like BCL6, the master regulator of the germinal center reaction, or BF2B, are expressed during most of this reaction. BCL6 is actually essential for the germinal centers to form, but need to be downregulated in order to allow cells to exit the germinal center and become plasma cells or memory B cells. And in fact, if you want to become a plasma cell, you will need to also upregulate specific master regulators such as the PRDM1 gene and the IRF4 gene in here. Okay, why is this important? It's important because if you look at these genes, each and every one of them is a common target of genetic alterations in B-cell malignancies. So perhaps one important advance that we made through all of this genomic analysis is that tumors are very smart, essentially hijack circuits or biological programs that are fundamental for the biology of the normal germinal center reaction, and then misuse them for their own proliferation and survival. So now I have said I'm going to focus the talk on diffuse large B-cell lymphoma, which is the most common of these cancers, and I'm sure you all know what it is. You also know that it can be considered curable in about 60% of patients that respond to currently available therapeutic approaches, but it does remain a significant clinical challenge because the remaining 35% or so of individuals who do not respond to treatments face a very dismal prognosis. So why is it that we cannot cure these tumors? And I think you will all agree that one major barrier to effectively treat these patients is the remarkable heterogeneity of these tumors, which can be appreciated from multiple standpoints. And in fact, the updated WHO classification of lymphoid malignancies now recognizes two major molecular subtypes of the disease, in particular in the DLBCL NOS group, which are defined by distinct transcriptional programs and reflect the origin from cells at distinct stages of differentiation. So the so-called GCB-like, germinal center B cell-like DLBCL, which share similarities with the light zone B cells, maybe a cell that is migrating in between the dark zone and the light zone. And then the less curable activated B cell-like DLBCL, which instead is presumed to be derived from a cell committed to plasma cell differentiation. Now, these subtypes are not only phenotypically diverse, they are also associated with distinct genetic alterations. And this was maybe one could think an outdated review, although I think these concepts are fully applicable as well, because what we found is that the genetic lesions associated with these tumors are in part shared across different subtypes, but there are also some that are preferentially or even exquisitely segregating with either GCB type DLBCL or ABC type DLBCLs, indicating clearly that these represent separate diseases and should be treated differently. In fact, as I mentioned, these two subgroups are associated with distinct clinical outcome. Now, this taxonomy has been further refined in more recent years, thanks to these seminal studies by the SHIP and Stouts laboratory that led to the identification of multiple genetic subgroups, as you see here, characterized by the presence of concurrent genetic alterations. And the potential clinical utility of these diseases is again underscored by the different survival outcomes that is associated with distinct genetic subtypes. So there is, however, a number of DLBCL that still don't get classified, but with the phenotypic characterization, also genetics may not explain all of them. So now that we have this more granular understanding of the germinal center reaction through the single cell studies, we asked whether this could inform further on the cell of origin on these tumors. So what we did was to develop an algorithm that would classify tumors based on expression modules that are associated with each one of these different subsets that we identified, and you see here on the left. And then we applied it to two large series of DLBCL cases, one from the NCI and the second one from the British Columbia Cancer Agency. And to make the story short, as you can see here, I would say maybe in part to our surprise, we were able to classify as many as 80% of these tumors, including some that had been assigned to the so-called unclassified COO class, which are the ones here in blue. And you can see highlighted in the white boxes the specific gene sets that are associated with each one of these populations. The other, I think, interesting finding that came out of these studies is that while you can see here in GCB DLBCL, the majority of them are transcriptionally similar to intermediate or light zone B cells. These compose maybe 80% of all cases, as we had and others had previously reported. There is a significant proportion of the activated B cell like DLBCL, which is this segment here in yellow of the pie, or also in the unclassified DLBCL that resembled quite remarkably the pre-memory B cell populations, suggesting that perhaps in a significant number of these tumors, the cell of origin is a lymphocyte that was committed to memory B cell differentiation, or perhaps an immature memory B cell. And then I think this is in line with some recent data from the Ari Melnik's group, who published this study looking at the TBL1XR1 gene, which is a gene typically associated to the so-called MCB, so to this subtype of ABC DLBCL, which also were preferentially pointing to a derivation from a memory B cells. So a cell that the mutations in particular would favor commitment to memory B cells at the expenses of plasma cell differentiation. Now, when you do these studies, obviously the question is always whether they could be of any clinical utility, that is, whether they could be associated with different clinical outcomes. So we looked at this in the relatively large number of cases that had clinical information. And to make the story short, you can look at all data in the paper. When we simplified this picture by essentially grouping different individual single-cell RNA clusters into major groups that comprise contiguous classes of single-cell signatures, there were maybe two groups that stood out, and this is include this light green group 4 here, which is associated with the signatures of intermediate, late intermediate cells or early light zone B cells, which had an extremely favorable prognosis, and this other group instead, group 2, that presents a fairly poor prognosis with overall mean survival less than 2 years. And this can be seen in both GCB type DLBCL and ABC type DLBCL, and was also highly reproducible independently in the two sets of data. Of course, these results await for validation in a prospective clinical series. And I think, however, that they were able to provide us a good number of candidate genes that may serve, could be tested as prognostic biomarkers for response to therapy or for overall survival. So now back to the genetics. As I mentioned, I think with over 2,000 cases that have been sequenced to date, for DLBCL, we've learned a lot about the genes that are disrupted in these tumors, at least in the coding genome. And so the bad news is that these are thousands of genes. So how do we deal with this complexity? And there can be multiple approaches, of course. I think that maybe all of them will be needed, but one way to reduce this enormous space of mutations would be by grouping them in a pathway-centric approach. So in biological programs or signaling pathways that could then represent potential actionable targets. And so a common theme that emerged from these studies, as we mentioned, is the highly recurrent targeting of genes that encode for histone chromatin modifiers, which are highlighted here by the arrow in these tumors, as well as in follicular lymphomas. Now, this is another common concept of biology that I'm sure you all know. Epigenetic remodeling is a key mechanism in the control of gene transcription, as well as in cell fate decisions. And so this dynamic and reversible state is maintained within the cells through the balanced activity of these macromolecular complexes that exert opposing functions in catalyzing the post-translational modification of the histone tails, and therefore change the chromatin conformation from a condensed and so tightly repressed state to an open chromatin configuration that is instead permissive for active transcription, as it provides accessibility of the underlying DNA to transcription factors. And so these macromolecular complexes include the so-called writers of the genome, acetyltransferases like P300 and KBP, and methyltransferases, including the KMT2D or MLL2 containing complex that typically counteracts the repressive function of the PRC2 complex. And then there are the erasers of the genome, histone deacetylases and histone lysine demethylases. So the tightly regulated machinery is disrupted and rewired in these tumors due to the presence of these multiple mutations. And you can see the numbers here. They are quite significant. If you put all of them together, probably all follicular lymphoma cases and a significant proportion of diffuse large B-cell lymphoma cases will have a problem with one of these genes, clearly indicating that perturbation of the epigenetic programming and reprogramming plays a fundamental role in the pathogenesis of these tumors. Now, the other key point that emerged from this genome sequencing analysis is that alterations in epigenetic modifiers, and particularly CREBP and KM2D, represent early events that are acquired by this putative common mutated precursor cell in early phases of the disease. And this is before the final clonal expansion, or in the case of follicular lymphoma, transformation to a more aggressive TFL, and typically diffuse large B-cell lymphoma, before the divergent evolution into either one of these diseases through the acquisition of additional genetic events. Now, the identification and targeting of these common precursor cells certainly represent a major area of investigation. I'm quite curious to talk with you and Sandrine and Bertrand about your view. It's an important area of investigation because, as you may understand, this would allow possibly not only to target and therefore eradicate the dominant tumor clone, should we be able to identify vulnerabilities in these cells, but also to eliminate this reservoir of early committed precursors that are responsible for relapse or transformation into the more aggressive DLBCL. So the mutations in CREBP and KMT2D are typically inactivating events that generate truncated proteins that lack these C-terminal catalytic domains, the Z domain or the histone acetyltransferase domain, and therefore would be functionally inactive. There are also a significant number of missense mutations, the one in green here, and particularly for CREBP, these include some highly recurrent hotspots, such as this arginine position 1446, which we had experimentally demonstrated also has a loss of function effect because it interferes with the affinity of this protein with acetyl coenzyme A, and therefore it also abrogates its acetyltransferase activity, but certainly will be important to understand whether there are also other consequences of these mutations on what are the non-catalytic functions of CREBP as we are learning in the field. So importantly, these mutations are monoallelic in as many as 60 to 80% of the affected cases, which I think it tells us two things, because the wild-type allele is almost invariably expressed. The first one is that probably cells require a certain threshold of acetyltransferase or methyltransferase activity in order to survive. The second is of course, the suggestion that these two genes may function as haploinsufficient tumor suppressors. And in fact, we and others have demonstrated this in vivo in animal models. Here I'm just showing a cartoon for CREBP, but this is a very similar scenario that we got when we modeled KMT2D loss in the mouse. And so the loss of these epigenetic modifiers alone promotes a hyperplastic phenotype in the germinal center, so would facilitate probably the accumulation of the additional genetic alterations as these cells represent the target structure for lymphomagenesis, but they are insufficient to drive full tumorigenesis alone. However, they were demonstrated as a bona fide tumor suppressor when combined with other genetic lesions that are also found recurrently in the human tumors. So in this case, with BCL2 deregulation, which synergize to promote the development of tumors with almost full penetrance, as you can see here, And these tumors display the phenotypic and genetic features of the human disease. In particular, here you can see expression of BCL6, B-cell markers, and also the evidence of somatically hypermutated immunoglobulin genes. So how do these two epigenetic modifiers contribute to malignant transformation? And here, to summarize a lot of work by us as well as others, what we know now is that these two enzymes both occupy a broad set of chromatin elements in the genome that display features of active enhancers or super enhancers. What was interesting, however, is that only a subset of the genes that are linked to these super enhancers are influenced and in fact are transcriptionally downregulated upon deletion of KMT2D or CREB-BP. And these genes, importantly, are enriched in biological functions that are relevant for the normal physiology of the germinal center reaction. You can see immune responses. There are many receptor molecules that are involved in interferon signaling, multiple components of the B-cell receptor and CD40 signaling pathway, both in KMT2D and also you see here in CREB-BP, as well as, especially in the case of CREB-BP, genes that are implicated in antigen processing and presentation, including C2TA and many of the MHC class 2 molecules. In addition, in the case of CREB-BP, we know that this is a protein that can acetylate also non-histone proteins. And so we had already shown years back that two important substrates for its acetyltransferase activity are the p53 tumor suppressor and the BCL6 oncogenes, which in fact have responded in opposite manners to acetylation. Acetylation is an absolute requirement for activation of p53, but instead modulates the suppression of BCL6 activity as it interferes with its recruitment of deacetylation complexes. And in addition, what we found is that if you look here, virtually all BCL6 known target genes are included among the regions that are occupied by CREB-BP, suggesting that these two proteins may control enhancer networks that are important in the germinal center light zone, and that their perturbed activity may, in addition, favor the unleashed function of BCL6. So upon deletion or reduced dosage of these molecules, cells may have problems in differentiation programs and may impair T-B cell crosstalks. Actually, this is a very important, certainly, area, I think, that we need to explore further, as this data suggests a link between the epigenetic disruption and the immune response to these tumors. Now, how can we translate this information into therapy? Now, chromatin modifications are reversible events. And so one way we thought could be to try and reestablish physiologic acetylation levels, since the second allele is often there. And this could be done by the use of histone deacetylases or histone lysine demethylases, although we know much less about this and about KMT2D. It is important that we identify probably more specific drugs, perhaps the HDAC3 inhibitors, part through a different mechanism that Ari Melnik has shown, but also importantly, we have to consider combination therapies, not as a single treatment, one candidate being BCL2 inhibitors, and obviously upon a proper patient stratification. But the other approach that one could consider is to identify dependencies that are created by the mutation in these cells and then exploit them for targeted therapeutic intervention. So in searching for candidate genes, we obviously considered as a promising protein the P300 acetyltransferase. So this is the paralog of CREB-BP. And not only these two proteins have a highly similar structural and functional features, but also these two genes are very rarely concurrently mutated, and definitely they are not biallelically mutated. There are no cases that we have seen where both genes have lost both alleles. So there were three questions that we wanted to address to verify that this could be a potential liability for CREBP defective cells. The first one, as I said, was to see whether P300 compensates, in fact, for loss of CREBP normal germinal center B cells. And if so, of course, we would want to know whether CREBP mutant DLBCL cells remain addicted to the residual HAT activity. And finally, should this be the case, can we exploit this dependency for therapeutic targeting? And so to address these questions, we generated compound mouse models for the conditional deletion of CREBP or P300, specifically in the germinal center, through the use of this Sigma-1 Cre deleter mice, and then monitor the germinal center responses in these cells. And the first observation here is that the loss of CREBP and P300 have distinct effects on these cells. In fact, you can see that not only they do different things, but they almost seem to go in opposite direction, with the CREBP knockout cells being germinal center B cells trending to an increase in these mice models, and on the other end, the P300 knockout animals are showing an almost twofold reduction in this population, indicating that P300 may in fact exert shared but also distinct functions on these cells. So to get some insights into the transcriptional programs that are responsible for these different phenotypes, what we did was to generate RNA-seq data from these models and then look at the differential expression. And then what you can see here, so are heat maps with genes that are differentially expressed between the CREBP knockout mice as compared to their control wild-type litter mates, with the P300 knockout germinal center B cells being shown as a default or as a reference. And then here is the reverse analysis for P300. And the point I want to make here is that you can see already that there isn't a strict correlation between the programs that are affected. In fact, with CREBP, we have again the genes I just told you that are implicated in these signal transduction programs as cells receive the signal to exit the germinal center, but this didn't seem to be significantly affected in P300 knockout mice. And on the other end, here you can see the reverse scenario. So there is a minimal overlap between the genes that are affected by these two genes, and this suggested to us that may be implicated in the control of different functions during the germinal center reaction. In fact, if you do a gene set enrichment analysis and look at what programs are overrepresented in these cells, here are again the famous players in a CREBP knockout cells, while P300 knockout germinal center cells were principally affected in genes that are implicated in cell cycle, DNA repair, as well as DNA replication or chromosome maintenance. And if you look at these programs, you will probably immediately associate them with different phases of the germinal center. And in fact, when we ran this gene set enrichment analysis with signatures that are upregulated specifically in these, I have to say, bulk populations at the time, you can see that certainly the programs that are pathognomonic of lysosome B cells are those that are preferentially lost in CREBP knockout cells, and the dark zone associated programs are instead downregulated synchronously in the P300 knockout mice. So now the question we wanted to ask, though, is whether the P300 gene compensates for CREBP, right? And so to look at these questions, we analyzed the compound mice, and to make the story short, here you can see that indeed double knockout mice are completely unable to form germinal center B cells. So you can see here the quantification from multiple experiments, and in fact it's important to mention that these few residual germinal center cells that we could detect in the double knockout mice is not shown here, but they represented escapees that did not efficiently delete both CREBP and P300 alleles. So I think this data obviously suggests that P300 is required for the survival and fitness of CREBP deficient cells. Now, these are normal cells. So the question is, what about the tumor cells? And here what we did was to generate a number of DLBCL cell models for the expression on inducible Cas9 clones. These are representative of the various states in which you find CREBP in the human tumor, so wild type, heterozygous, and also one of the rare biallelically mutated CREBP cell lines, and then we transduce them with guides to target specifically P300 or a new drug control region. Now I'm going to show you only one piece of data, which was a competition assay where we would then mix in 50-50 ratio these cells, induce the deletion of P300 by doxycycline, and then monitor the fitness of these cells by FACS analysis, looking at the ratio of RFP versus GFP. And what you can see here is that as long as you don't treat with doxycycline, these cells grow in a quite comparable manner. However, the deletion of P300 upon doxycycline treatment led to a significant and dose-dependent, I would say, in that was a proportionally more marked manner as we moved from the wild-type cells to the heterozygous CREBP cells with the maximum effect in the CREBP homozygous mutant cells, clearly indicating that CREBP mutant cells are counter-selected and require P300 as for their survival. So can we exploit this dependence for therapeutic targeting? And to this end, we use these isogenic DLBCL clones that are either null or express normal levels of CREBP, and took advantage of two molecule inhibitors for CREBP P300s. There are no specific inhibitors that are only targeting P300, but these are quite specific for this class of acetyltransferases. And one is a HAT inhibitor, and the other one is a molecule that targets the single bromodomain shared by these two proteins. And to make the story short, you can see here when we treated this isogenic zone, both compounds had a preferential toxicity in the CREBP knockout cells in red as compared to their isogenic wild-type clones, indicating that there may be a therapeutic window for the use of these drugs. One of these is actually currently in clinical trials, and also I think providing the basis for the development of drugs that could specifically then inhibit P300. So the model we would like to propose is that by enlarge P300 and CREBP exert both shared and distinct functions in the germinal centers. We think that the normal cells would obviously take advantage of these common targets for maintaining their fitness, while specifically modulated programs could be the ones that tumors hijack for their own survival, including, again, those that modulate interaction with the microenvironment or differentiation. But these common targets are the ones that could establish synthetic lethal interactions, and that we could exploit this therapeutically as CREBP wild-type cells because of this redundancy would have limited toxicity, while these compounds, as we've shown, can be preferentially toxic to the CREBP mutant lymphoma cells that are addicted to P300. And now I hope I have a few, 5 or 6 minutes to tell you instead some unpublished data that we have obtained recently of a novel functional interaction between CREBP and KMT2D. There were several observations in support of such interaction. The first one is genetics, because as you can see here, as many as 50% of follicular lymphoma cases and a good proportion of diffuse large B-cell lymphoma cases, in fact, over 30% of the so-called EZB or C3 types of GCB DLBCL carry concurrent mutations in these two genes. The second is their co-occupancy on chromatin, because this is significant, as you can also see from these plots that show the relative distribution of CREBP peaks as compared to the closest KMT2D peak and vice versa in here. And finally, it has been shown that KMT2D is required at enhancers and super enhancer for optimal CREB-BP recruitment and transcriptional activation. So what we wanted to ask is whether CREB-BP could in turn regulate KMT2D by direct protein acetylation. And so to address this question, we first isolated normal germinal-centered B cells as well as naive B cells from reactive tonsils. So we treated them for 2 hours with TSA niacinamide, which are known drugs inhibiting the acetylases. And then we analyzed these cells by Western blot analysis for acetylated lysins following immunoprecipitation with the KMT2D. And as you can see here, first of all, both naive and terminal center B cells were efficiently immunoprecipitated with KMT2D antibodies, and both of them showed a strong signal when we applied the antibodies against acetylated lysins. You can see here also this other component of the COMPASS complex documenting that acetylation of KMT2D represents and has a physiological role in germinal center B cells. And very similar results were obtained by looking at DLBCL cell lines that are wild type for KMT2D and CREB-BP. So you can see here again that in basal condition, there is no acetylation that can be detected, very little, but the addition of TSA or niacinamide led to the significant increase in this modification, which was maximal upon co-treatment. And so we think that both class 1 and class 2 and 3 the acetylases are implicated in this modification. Now, the question is whether CREB-BP is the acetyltransferase, right? So what we did here is to use an overexpression system where we co-transfected in 293 T cells, either flag tag CREB-BP proteins or HA tag KMT2D proteins, and then we looked by, again, Western blot and after immunoprecipitation at acetylation of KMT2D. And you can see already here in the input that when you transfect KMT2D alone, that there is no signal detected by this antibody, but when the two proteins are co-transfected together, there is a very clear band here of the expected molecular weight for KMT2D. And indeed, this is KMT2D as demonstrated here by the analysis in immunoprecipitate with the specific antibodies. We also showed that this modification requires the enzymatic activity of CREB-BP, as we were completely unable to detect it when we used these two mutants of CREB-BP that actually recapitulate what we see in the human tumors. One is this hotspot missense mutation at arginine 1446, and the other one instead is a truncated protein that lacks the HAT domain completely. And you can see these are both unable to self-acetylate themselves and completely unable to acetylate KMT2D. So CREB-BP enzymatic activity is required for this activity. Now, to unequivocally document that this is not the result of an indirect mechanism, what we did was to perform an in vitro acetylation assay with semi-purified KMT2D or CREB-BP protein. And so again, here when we put in the tube only KMT2D, even in the presence of acetyl coenzyme A, there is no signal detected. However, the addition of increasing doses of acetyl coenzyme A in the presence of CREB-BP led to a dose-dependent augmentation of this signal. And this was not true when we use the CREB-BP missense mutant that is enzymatically active, indicating that CREB-BP can indeed acetylate KMT2D directly. Now, there are multiple mechanisms and in particular that could be affected, or consequences that could be dependent on this post-translational modification. We are exploring them in the lab, but today I'm just going to show you one piece of data done almost to suggest that perhaps acetylation of KMT2D may interfere with its ability to methylate its substrates. So here we have again wild type and knockout isogenic clones that lack CREB-BP. you can see that the total amount of KMT2D is not changed, but there is a significant reduction in acetylated KMT2D. And now if you look at the chromatin extracts from these same cells, as expected, there is reduction in K18 acetylation and K27 acetylation, but also we saw a measurable diminishment in the K4 monomethylation, and you can see the quantification down here. Now, this is not a complete abrogation, but I think that they obviously could be due in part to redundancies, both for CREB-BP and for KMT2D. Because we cannot use genetic deletion of both CREB-BP and P300, which is incompatible with life, what we did was to take advantage of these inhibitors. And now, again, you can see that TSA niacinamide favors the enhancement of this acetylation signal. But when we abrogate the activity of both acetyltransferases, this is completely lost. And again, there's also a marked reduction of the monomethylation signal. So perhaps one effect of acetylation could be to facilitate the enzymatic activity of KMT2D. Finally, we looked at whether haploinsufficiency of CREB-BP and KMT2D could cooperate in lymphomagenesis, again by resorting to mouse models and specifically deleting these genes in germinal center B cells. And this is one representative piece of data. We're looking at germinal center B cells again with specific markers. You can see that as we and others had reported, the single heterozygous deletion of CBP or KMT2D did not have major effects in this population. However, the combined loss of these two proteins was associated with a significant increase in the germinal center B cell population, which is quantified down here, and was statistically significant. So we suggest that CREB-BP loss combined with KMT2D haploinsufficiency may in fact facilitate the expansion of the common precursor cell and perhaps allow it to remain in circulation for years, therefore acquiring or having the possibility to acquire additional genetic events until this would drive a final malignant transformation. And so to summarize what I told you today, in the second part of the talk, KMT2D is acetylated by CREB-BP in both normal and transformed germinal center B cells. We have shown that loss of CREB-BP-mediated KMT2D acetylation is associated with this reduced K4 monomethylation, which may suggest a role in modulating the activity of KMT2D. And also, the CREB-BP-mediated acetylation of KMT2D may suggest that tumors with CREB-BP defects would also have double haploinsufficiency, and maybe this is particularly relevant in the context of the common precursor cells. So the question is whether then CREB-BP targeted epigenetic therapies may be effective in KMT2D mutant tumors. And with this, I'll stop, obviously not without thanking the many colleagues, postdocs, and students that have contributed to these studies and to what I've shown you. This is a work of many years in my groups, as well as in the group of Riccardo Dalla Favera and Katia, who have been collaborating together for many years, as well as you can see here many external collaborators. And thank you very much for your attention.
Thank you very much, Laura, for this amazing presentation. Very interesting, very clear, very complete. So I will perhaps start with the first question that I think is a question from Bertrand. So it's more a general question, in fact. The question is that many mouse models show dramatic transformation with relatively short latency with just a couple of alterations, whereas in human subtypes, there is hundreds of alterations. Do you think it is necessary to have all these alterations, or do you think that there is accumulating by bystander effect how are the early epigenetic mutation acting or pushing transformation of pretumoral GCB cells during the primary GC visit versus chronic reentries? So how could you recapitulate in your model this idea of a lot of additional genetic alterations, whereas in mice, finally, with 2 or 3 alterations, you have a lymphoma that developed?
Well, but with mice, I'm sure that Sandrine will agree. So with mice, first of all, it's always an artificial system because we are deleting the gene in each and every cell. So obviously humans would have stochastic occurrence of these mutations. And I'm sure that then the reservoir of cells that could undergo malignant transformation would obviously in this case make facilitate a higher frequency of lymphoma developing. I think that indeed we need, there's no doubt that tumors need multiple alterations to become a tumor. And in this context, epigenetic modifiers probably have a little bit of an atypical role as tumor suppressors in the fact that they are not sufficient alone to promote full malignant transformation. So perhaps I think, and this is pure speculation, but the function of these lesions that early during the ontogeny of a tumor would be simply to maintain a certain plasticity of the epigenome and probably in a mild manner that is compatible then with long-term life and maybe recirculation through multiple germinal center reactions, which would favor the accumulation of additional lesions, favor lymphomagenesis. So probably both the interference with differentiation, which may maintain cells and this environment, or maybe even interferes with differentiation of memory B cells to plasma cells, would have a role. And definitely, I think there are already evidences from your work, Karin, as well, that epigenetic damage by interfering with the expression of important receptor molecules that are implicated in the crosstalk with T cells or follicular dendritic cells may shift the dependency of these germinal center B cells from one to another and ultimately allow them to evade this default death program of the germinal center, or later on to evade maybe tumor cell recognition.
Thank you. I think we have another question from Cedric, University of Rennes, who asks, are QABP and EP300 redundant or complementary?
I think probably both. They are certainly redundant in that you can, I think they're certainly redundant in part because you can, you can still survive, at least in the context of germinal center B cells. Of course, the context matter. So developmental stages are different, but in the context of germinal center B cells, we have seen that QABP can be deleted without dramatic effect. And on survival, but if you take away P300 as well, that would be incompatible with life. So I think there's no doubt that one is represents an essentiality in QABP mutant cells. At the same time, as you've seen, that there are distinct functions that are modulated by these genes. So I think that in the context of a normal germinal center reaction, it will be interesting to see how these two proteins can modulate different substrates or different targets. And unfortunately, what we have done so far is only looking at transcription, right? But the CBP, P300 also acetylate non-histone proteins. So there could be compensatory mechanisms also at that level that we have not seen, maybe hyperacetylation of other proteins or that they could compensate for the loss of individual proteins.
Is there any difference in your models between the time of development of follicular lymphoma when you compare QABP or EP300 or the combination of both, despite from the frequency of lymphoma? Do you have difference in terms of timing of development of the disease?
I cannot answer this question because we did not study tumor development in the P300 knockout mice. I mean, there are studies that have been published in the old times, right, where deletion of P300 was also associated in part to tumorigenesis in other cellular contexts, but we have not done that. So,
okay.
I don't know.
Okay. So there is a question regarding the mice model that deplete both KMT2D and QABP constitutively. Do you think an inducible system to see the kinetic and the impact of these two factors, maybe in terms of kinetics, QABP first and KMT2D second, or something like that? Have you?
Can you repeat the question? Because you mentioned the word constitutively.
I think you use a CD19 or in your double.
We use both, but I was showing you the sigma 1 CRE. Yes, okay. But anyway, yes, that's. And the question was?
Don't you think to use? But in fact, you use an inducible system, because if you use sigma or CD19, it's an inducible system, but. It was, I think the idea was, do you have tested to maybe to have a QABP first and KMT2D second using several tweaks to maybe delete the first one and then delete later the second one to see how they cooperate and into the
we have not done that. That's what I hope we may be able to do. These are big experiments, and I think that in fact we should look at that, because the timing of these lesions will most likely be important also in the kinetics of tumor development. It would also be important to see whether, I mean, obviously have inducible systems to see whether you can, at least in the context especially of compensatory mechanisms, to see whether you can make cells, then you can, reconstitute the cells and then modulate the tumor behavior by playing with this tumor suppressor. But I think that, I mean, I'm not sure, they may not even have a role in tumorigenesis once the tumor is established.
Could be.
There is another question from Michel Cognier, so a naive question. He says, is the KMT2D deacetylation pathway also known? Any potential additional regulation thereof?
I think we don't know anything. We don't know anything about the acetylation and the deacetylation. I mean, the acetylation now is a nice new finding, but there's a lot of work to be done.
And also regarding this acetylation or the modulation of KMT2D by QABP, so why, if QABP is able to modulate the activity, methyltransferase activity of KMT2D, why do we need to lose KMT2D in the lymphoma cells? Because if QABP is able to modulate, maybe we won't need to lose it. And most of the time we lose it probably secondarily of QABP. So I don't really, do you have any input on that?
So I don't have an answer yet. Yes. And obviously that's an important question. I think one would have, I mean, it all depends on what is exactly the mechanism and what is the function that is of KMT2D that is influenced by QABP mediated acetylation, as this could be, interaction with the specific proteins. It could have to do with, in fact, maintaining certain functions while eliminating others. And it is true that there is a good proportion of cases that have biallelic deletion, but I think maybe 70-60% of DLBCL for sure we would not have lost the second KMT2D allele. So in that respect, perhaps acetylation could take care of certain functions of the second allele, perhaps maintain certain, abrogate specific functions that are useful to the tumor while maintaining other important roles for these epigenetic modifiers that are important for the, for all cells to survive for many functions.
That might explain, Laura, as well the kinetics as well. That is
yeah,
quite
exactly. In fact, I think that this is.
Yeah.
I think that it would be interesting because if, as I think now is quite clear that the KBP mutations come first, then yes, then maybe that could have yet a maybe more modest or subtle effect in modulating whatever aspect of its activity is important in the cell.
Can we have a last question, Karine and Sandrine, because unfortunately we're going to have to close the session soon. One or burning question?
I don't see any.
I think you already answered this question, but
yes, I think this is the same one.
While you search, I'm burning to ask the question of the microenvironment, which I was sure you would be asking, Karine, but you didn't.
Yes, I didn't, because I'm sure that I will not have the full answer. So there is so many questions, but so for sure we are discussing a lot about DLBCL today. But even in DLBCL, the microenvironment is crucial. It is not only a question of follicular lymphoma, for sure. So do you have any idea, at least at the organization level, without discussing very specific aspects of T-cell regulation and so on, but in terms of organization of the germinal center, of the light zone versus the dark zone, at least at this very basic level, do we have any comparison of your different mice models and how it could impact the organization of the whole lymph node and GC reaction?
I don't have an answer in terms of data, but I don't have an answer, unfortunately. But yeah, these are the experiments that need to be done, I think, and the single cell approach will be helpful, I think, in this context, because obviously it may pinpoint to the populations, not only in the B lymphoid cells, but also in the accessory cells.
A single cell RNA-seq, but also probably in situ analysis.
Yes, of course. Yeah, oh, absolutely.
You missed a lot of cells probably, but okay, that's all.
Yeah, absolutely, absolutely. I that's even more important maybe, but I thought
no problem.
Combine.
Okay.
Okay. So I think we need to close the session. Unfortunately, that's the problem with the webinars. They have to stop. Sometimes more formally than the real session. We all would like to have you live, Laura, but it was a very fascinating talk. Thank you many times for being here today with us for the Red Talk that I'm sure will continue. Don't forget that for the audience that you can continue asking your questions after the closing of this session. Laura will stay a couple minutes to keep on answering your questions, so use the question tab. I just wanted to say many thanks to all of the audience that were here tonight. Stay tuned for the next Red Talk that will be beginning of January. It will be about MCL, and it will be an educational session with Morgane Cheminant in Paris and David Chiron in Nantes. And I think I don't know if I have to wish you a good evening, a good morning, a good night, but wherever you are in the world, thank you very much for being here and attending the Red Talks. Bye-bye.
Thank you, Bertrand, and thank you everybody, Sandrine, Karine. Thank you very much.
Thank you.
Thank you,
Bye-bye.
Laura.